Hello, my name is LeoChan, and I'm the APAC manager for the Science and Technology Advisory team at 10x Genomics. Today, I would like to talk about how you can break your research barrier in spatial biology using our 10x Genomics Visium Spatial solutions. 10x Genomics Visium Spatial platform was based on the technology initially acquired from spatial transcriptomics in 2018. We have been honored to see Visium features in the covers of Nature Methods for its Methods of the Year and the Scientist's Top 10 Innovations last year in 2020. Since our products was released, we have seen a tremendous adoption by the basic and translational researchers in various application segments. But first, why spatial profiling analysis? Here, we are looking at a human breast ductal carcinoma in situ tissue sample. Its morphology on your left already gives away its complexity and heterogeneity. So what is the best way to uncover the tissue's complex environment? RNA analysis has certainly evolved as a great analyte for validation and for discovery research. Low-plex analysis using several single gene markers might be a good start, but this is limited. In the best case, you will need to study the whole transcriptome with spatial organization, enabling new and unbiased discovery. And this is exactly what Visium enables. For you to retrieve complete and unbiased biological informations, while at the same time to retain the organizations of the tissue environment. Here on your right, different colors in the spot represent different gene expression patterns based on thousands of RNA markers. Our Visium Spatial Gene Expression Assay on your left is compatible with SNAP frozen and OCT embedded tissue. And the Visium Spatial Gene Expression for FFPE assay on your right is compatible with FFPE samples. For fresh frozen samples, you can perform whole or targeted transcriptome analysis. For FFPE, we are utilizing whole transcriptome probe panels for mouse or human to enable spatial gene expression analysis. With Visium, you can link the transcriptome to the H&E stain image, or you can also link the transcriptome to immunofluorescence stain image to co-detect protein and RNA. At 10x, we offer integrated software tools that you can download from our website for data analysis and visualization. Before I proceed further, let's take a quick look on how Visium works. At the end, the Visium is made up of two kinds of data: histology or immunofluorescence from a microscope image, and the gene expression data from sequencing. The power in the Visium solutions is to combine these data. Here you can see that we have combined the histology image with the spots that are coming from the gene expression slide. Each dot represents one of the spots on the slide surface. You can approach analysis in several ways. You can start with the histology, perhaps to enlist a pathologist to help you identify regions of interest. And then you can overlay the gene expression data to look for differential gene expression signatures or marker genes in your regions of interest. On the flip side, you can start with the gene expression data, looking at your favorite genes, and then explore the clustering results for interesting gene expression signatures. Then add on the tissues and see where your genes and clusters of interest are in the context of the tissue. Our Visium solution is being used in many different applications. In oncology, immunology, neuroscience, developmental biology, pathology, and many others. Next, I'm going to talk about some examples through publications by our users. First, let's look at two examples in oncology. The first publications that I would like to discuss is from researchers at the University of Tokyo and other collaborating institutes in Japan. This work was published in Communication Biology this year, 2021. Many cases of ductal carcinoma in situ, or DCIS, of the breast, they will never progress to invasive ductal carcinoma, indicating that they are overtreated under the current criteria. Although various candidate markers are available, the relevant markers for delineating the risk categories have not been established. In this study, the authors analyzed the integrated clinical features of more than 400 cases of ductal carcinoma in situ and performed deep sequencing analysis in a 21-case discovery cohort and in a 72-case validation cohort. They identified the most critical markers for the aggressiveness of DCIS. One of these markers is GATA3 mutation positivity. Here, we are looking at the spatial transcriptomic analysis of a patient donor with GATA3 mutation positive DCIS. The H&E stain are on your left. Morphologically, cells are classified as cancer cells in situ or non-cancer cell. 3 cancer cell populations are shown with different colors as indicated in this middle left panel. 4 non-cancerous regions are also indicated. On your right, the spots in which the GATA3 mutations were detected are colored red. In the spots with GATA3 mutations, they are mainly enriched with signatures for epithelial mesenchymal transitions, or A, and angiogenesis, or B. The spots without the GATA3 mutations are mainly enriched with signatures for tight junctions, estrogen response, that is C, and mTORC1 signaling. Overall, the authors use spatial transcriptomics, and they also do single-cell DNA-seq to reveal that GATA3 dysfunction upregulates epithelial mesenchymal transitions, invasions, and angiogenic pathways, followed by progesterone receptor downregulation. These results reveal the existence of heterogeneous populations of DCIS and provided predictive markers for classifying DCIS and optimizing treatment regime. The next publication in Cell last year is by researchers from Stanford University in the US. As shown on your top left, Their experiments start by generating 10x chromium single-cell gene expression data sets from normal skin and squamous cell carcinoma clinical samples from patient donors. The authors then integrated our Visium datasets, and they also conducted multiplex ion beam imaging, a technique they use for protein detection. Additionally, they also performed CRISPR functional screens for validations. One of the highlights of this publication is that the authors characterized the tumor microenvironment, and they identify a cell populations which they call tumor-specific keratinocytes, and this is shown on this bottom left, or the TSK. And these cells are very different from the epithelial cells. They express cascades that indicate invasive behavior and cell movement. Moreover, the authors identified the spatial locations of these cells. They resided in the fibrovascular niches. That is, these cells possess the property to invade and move as inferred from the gene expression data. And they are strategically placed close to blood vessels for getting into the circulation. These tumor-specific keratinocyte cells reside in the tumor edges, as shown in these spatial data sets. And their partners in crime are the endothelial and cancer-associated fibroblast cells that they infer from the cell-cell interaction analysis from the spatial data. All of these plays important roles in tumor progressions. Taken together, their data define the cellular compositions and architecture of cutaneous squamous cell carcinoma. They examine the spatial niches where they interact from the spatial data, and they also examine the communicating gene networks that they engage in cancer. What is unique about the publications I just talked about, and many more that I'm going to talk about soon, is the integrations of a chromium single cell and the Visium spatial gene expression data. With these highly complementary techniques, you can look at the what, the where, and the how to level up your biological insights. At 10x, we offer the chromium single cell and the Visium spatial workflows with these complementary products. For immunology, I would like to talk about an examples on viral infections of the lung, which also integrates single cell and spatial transcriptomics technologies. This publication in Nature last year is by researchers at the St. Jude's Children's Research Hospital in the US. The lung extracellular matrix, or the ECM, provides structural support that is critical for lung function. And it also provides signals that coordinate immune responses. Many lung damages induced by viral infections are the results of these infiltrating immune cells, which can kill both the infected and bystander cells. Because of this, some components of the ECM could be promising therapeutic targets. Here, the authors look at what is likely to be a delicate balance between pathogen clearance and immunopathology. They use single-cell and spatial transcriptomic technologies to unravel the role of the lung stroma cell populations in coordinating whole responses to respiratory infections. They sorted lung cells from mice at 0, 1, 3, and 6 days after infection with influenza A virus. And they first used single-cell gene expression profiling analysis to identify all the distinct cell populations. As indicated on your lower left, within the fibroblast group, they observe multiple transcriptional states. To gene expressions, they identify them as resting, ECM synthesizing, or inflammatory. When they look at these inflammatory fibroblasts over the entire course of the infection, they found these two distinct activation states. The number of these damage-responsive fibroblasts increased over the entire course of infection, reaching a peak of around 12 days. The interferon-responsive groups emerged by day 3 and then gradually decreased. They also identify one ECM protease, ADAMTS4, which shows significant upregulation in fibroblasts after viral infection. The authors then use spatial analysis to look at how the fibroblasts are organized at 10 days post-infection. This is during the peak of the respiratory distress. They found that the signature genes associated with the damage-responsive group, including ADAMTS4, are localized to the area of the interstitial inflammations. And you can see this in the right panels as the red group 0. But these are absent in other regions. Their data placed these damage-responsive fibroblasts expressing ADAMTS4 in the distal airways as a critical inflection point where the hosts either begin to recover or their conditions become worse. For neuroscience, let's look at an example published in Nature Neuroscience. These publications this year in 2021 by researchers from the Johns Hopkins in the US, they examined the human dorsolateral prefrontal cortex from fresh frozen postmortem brain of neurotypical donors. By using histology, graph-based clustering, and layer marker genes, and through spatial technology, these authors were able to annotate the 6 layers of the cortex and identify the extensive layer and rich expression signatures and redefine associations to all the previous markers. I want to highlight that they integrated spatial resolutions to their single nucleus RNA-seq datasets generated from human brain tissue to provide further insights about the functions of molecularly defined cell types. Specifically, the layer and rich expression profiles and differentially expressed statistics derived from the enrichment model in their Visium data can be used to spatially register their single nucleus RNA-seq datasets and add on the layer and rich informations to data-driven expression clusters that do not contain inherent anatomy. And this is schematically shown on the left. Overall, their analysis demonstrated how spatial integration framework can be readily applied to any existing single nucleus RNA-seq or single cell RNA-seq datasets to add back the anatomy. For developmental biology, I would like to talk about an example studying intestinal development. In these cell publications by researchers from the Oxford University in the UK, they really make some huge strides in understanding the development of the human intestines. They were able to capture human intestinal tissue samples with a full thickness, which enabled them to map all the compartments. They started with more than 70 samples collected from 17 individual human embryos from a range of developmental stages and different locations. First, they used 10x chromium single-cell gene expression solutions, and they clustered cells into 9 different compartments, and also into 101 subcompartments based on the transcriptional signatures. They then examined 8 tissue sections from 5 samples ranging from 12 post-conceptual weeks all the way to adulthood. They used the single-cell RNA-seq atlas that they generated as a reference And then they carry out factor analysis to determine the likely single cell composition for each of the spots on the Visium slide, thus spatially localizing all the single cell RNA-seq clusters on the Visium data. This is schematically represented on the right. They then also created a map of genes involved in several pathways to clarify where molecules, which are known to form gradients important in morphogenesis, how they act in space and time, and to direct sequential waves of cellular differentiation. For cardiovascular research, let's look at an example studying heart attack. Myocardial infarctions, MI, or heart attack, is the leading cause of mortality worldwide. While advances in the acute treatment have been made, the late-stage mortality is still high, driven by an incomplete understanding of cardiac remodeling processes. Cardiac remodeling after MI involves immune cell recruitment and demarcations of the infarcted area, followed by tissue digestions, phagocytosis, myofibroblast activations, scar formations, and the formations of new vasculars. Thus, an understanding of the exact cellular and molecular mechanisms of cardiac remodeling processes will be the key to develop novel therapeutics. Here, the authors from the University of Oxford in the UK, they combine single-cell gene expressions in chromatin accessibility technologies and spatial transcriptomics to study the cell type-specific changes in gene regulation. They profile different physiological zones and time points of human myocardial infarctions and human control myocardium to generate an integrated high-resolution map of cardiac remodeling. This approach allowed them to increase the spatial resolution of cell type compositions and provide spatially resolved insights into the cardiac transcriptomes and also the epigenome. They were able to identify distinct cellular zones of injury, repair, and remodeling. The border zones of the myocardial infarctions is of particular interest, since spatial remodeling of this area is linked to the recovery of the cardiac functions. As shown on your this upper left, despite the homogeneous H&E staining and the transcript distributions across the Visium slide, the authors observed extensive heterogeneity spatial gene expression profiles, and they identified a specific spatial transition zones in the center of the specimen around the cluster 1 area. This zone separates distinct areas of gene expressions in the upper left part from the lower right part of the specimen. Overall, their analysis indicated spatially distinct gene expressions and regulations in the border zones with cardiomyocyte subsets that are specifically spatially located with distinct regions of injury, inflammations, and remodeling. Concluding remarks. As concluding remarks, I want to articulate that the Visium is more than just a tissue slide. You can perform unbiased whole transcriptome analysis in a morphological context to discover new insights into normal and diseased tissues. For fresh frozen samples, you can combine the critical insights provided by spatial transcriptomics with the ease and breadth of targeted panels for a more focused gene expressions analysis. You can visualize spatial patterns of gene expressions together with protein detection by immunofluorescence on the same tissue section to gain a new perspective on tissue complexity, linking protein, and gene expression. You can utilize the standard fixation and staining techniques alongside gene expression and protein detection to unravel the biological architecture. And you can bridge digital and molecular pathology with in situ gene expression to visualize biological signatures, identify different predictive markers, and differentiate normal from diseased tissues in all of your regions of interest. The publication examples that I talk about today represent a small portions of use cases that have been published. For more information, including finding more papers, example data sets, protocols, technical notes, and application notes, please visit our website at 10xgenomics.com. Thank you.